Welcome to this explainer. Today we're diving into some serious historical detective work to uncover exactly how scientists managed to categorize every single living thing on Earth. It's a fascinating story of trial, error, and some absolutely massive biological breakthroughs. Let's get into it.
Imagine trying to organize a messy, cluttered room. Hard enough, right? But in this case, the room is literally the entire planet, and the items scattered around are millions of wildly different biological species. Where do you even begin? How on earth do you create a functioning filing system for life itself without it devolving into total chaos? Well, let's rewind all the way back to Aristotle. Known today as the father of biology, Aristotle actually kept things surprisingly simple. While he did look at plants, his main focus was really on animals. He divided them up by where they lived, water, land, or air, and by a very basic physical trait, whether they had red blood, which he called anima, or white blood, which he called anonyma. It was a great start for sure, but way too simple for the immense complexity of life on Earth. But, as microscopic technology evolved and our field of vision blew wide open beyond the naked eye, our understanding of life completely transformed. This kicked off a centuries-long puzzle to build a better system. We're going to track this evolution across four major leaps, moving from a basic two-kingdom system in 1758 all the way to the famous five-kingdom system in 1969. Enter Carl Linnaeus in 1758. He established a super strict two-kingdom system consisting of Plantae and Animalia. And his criteria for this division were incredibly straightforward. He based it entirely on two things: locomotion, so could the organism move, and its mode of nutrition, meaning how did it get its food. But here is the catch. While Linnaeus' system was definitely elegant, it had some massive blind spots. His system completely failed to distinguish between simple prokaryotic cells and complex eukaryotic cells. It didn't separate single-celled organisms from multicellular ones either, and perhaps most awkwardly, he just sort of shoved fungi in with the plants, which biologically makes very little sense. To solve this glaring cell issue, Ernst Haeckel stepped up to the plate in 1866. He introduced a third kingdom, which he called Protista. This was created specifically to house those tricky unicellular organisms that just didn't fit before, things like bacteria, yeast, amoebas, and blue-green algae. So now we had Protista for the single-celled stuff, Plantae for multicellular organisms with cell walls, and Animalia for the ones without. But as we know, science is all about constant refinement. Haeckel solved one issue, but accidentally created another. Because of the criteria he used, he was still grouping totally different basic cell structures together. Prokaryotes and eukaryotes were literally sharing a kingdom. On top of that, various types of fungi were now scattered chaotically across these different kingdoms. Fast forward to 1956, and here comes Copeland, bringing us a four-kingdom classification. Copeland recognized the fundamental difference in cell architecture, so he added a brand new kingdom called Monera. This was specifically designed to isolate the simple prokaryotic cells, like bacteria and blue-green algae. That meant Protista was now strictly reserved for eukaryotic unicellular organisms. Much cleaner. Yet, our historical detective work still wasn't quite finished. Copeland's new system was vastly better, no doubt, but it left one major outlier causing taxonomic chaos. Where on earth do the fungi actually belong? The biological contradiction here was glaring. Fungi, like your everyday mushroom, were still being grouped alongside plants. But plants are photosynthetic autotrophs. They make their own food. Fungi are a totally different ballgame. They're heterotrophic saprophytes, meaning they literally derive their energy by feeding on dead and decaying organic matter. They absolutely did not belong with plants. This ultimate taxonomic puzzle was brilliantly resolved in 1969 by Robert Whittaker. He introduced the five-kingdom classification system, finally giving fungi its very own kingdom. He justified this separation because fungi have a totally unique cell wall made of chitin, rather than the cellulose you'd find in plants, along with their distinct heterotrophic mode of nutrition. When you lay all five kingdoms out side by side, you can see exactly why Whittaker's system succeeded. He didn't just guess, he used strict observable criteria. By looking at cell structure, organization, mode of nutrition, and evolutionary relationships, he cleanly and logically divided life into the five kingdoms we recognize today: Monera, Protista, Fungi, Plantae, and Animalia. It brought a profound sense of order to biology. Okay, so now we finally have the major kingdoms sorted out, but scientists needed universal rules to actually file newly discovered organisms into them. You can't just toss a bug into a kingdom and call it a day. You need a precise system. To do this, biologists rely on two heavily interconnected branches of biology, systematics and taxonomy.
Let's define these terms, starting with taxonomy, a term given to us by Candolle. Taxonomy is the branch of biology that deals strictly with identification, nomenclature, which is just a fancy word for naming, and classification. It essentially gives us the foundational framework, the literal filing cabinets for organizing all these organisms.
Then we have systematics, a term coined by our old friend Carl Linnaeus. Systematics takes that rigid taxonomic framework a massive step further. It still deals with identification, naming, and classification, but it also studies the deep evolutionary and phylogenetic relationships between these life forms. It doesn't just ask, what is this? It asks, how is it related to everything else over time?
So using these tools, how do we practically file a newly discovered creature within this massive global system? We use what is called the Taxonomical Classification Hierarchy. We cascade down a strict 7-level hierarchy. We start at the very top, at the broadest category, the kingdom. From there, we filter down through phylum or division, then class, order, family, genus, all the way down to the highly specific species level. Each step you take down gets more and more exclusive until you are looking at one exact, unique type of living organism. Once we hit rock bottom, that final, specific species level, we have to name it. And we do this using the binomial system of nomenclature. This was yet another absolute masterstroke by Carl Linnaeus. It ensures that a scientist in Brazil and a scientist in Japan use the exact same name for the exact same organism. It completely avoids linguistic chaos. Now, this universal naming code relies on a few strict, unbending rules based on the genus and the species. First, the names have to hit that sweet spot of being between 3 and 13 letters long. When typed out, they must be in an italic font, or if they are handwritten, the genus and species must be underlined separately. And most importantly, the first letter of the genus is always capitalized, while the first letter of the species is always lowercase. Let's look at a really delicious example to make this concrete. The common mango. Its scientific name is Mangifera indica. Mangifera is the genus, starting with a capital M. Indica is the species, starting with a lowercase I, and they are cleanly underlined separately. Thanks to this simple rule, scientists globally know exactly what plant is being discussed. And oh, here's a really fun fact hidden in these rules. If you happen to be the lucky scientist who discovers a new species, your name can actually be added at the very end of this binomial tag. If your name is added, it sits right at the end, and the first letter of your name is capitalized. It is honestly the ultimate scientific flex. This centuries-long historical problem-solving journey, from Aristotle's simple blood tests to Whittaker's 5 kingdoms and Linnaeus's naming systems, brought incredible order to what was once biological chaos. But it leaves us with one massive, provocative question. As our genetic technology improves, as we map more DNA and uncover even deeper, previously invisible evolutionary relationships, will Whittaker's 5 kingdom system survive the test of time, or is a brand new 6th kingdom waiting just out of sight, ready to rewrite the textbooks all over again? Thanks for joining me on this explainer. Keep asking those big questions, and I'll catch you on the next one.